Hi, I'd like to give you a quick rundown on how to use the Waever data integration platform from Applied Exomics. This is a free platform for you to use. We can use it in client projects to tailor the underlying data to in-house data or change the annotations and information that you see in the tool. But what we provided here is a useful standalone platform for exploring proteins that you're interested in alongside their interaction partners in terms of biological networks, the functional relationships within those networks, and all of that within a cell type or tissue specific context based on whether or not the genes that encode those proteins are expressed in different tissues. So this will be a quick click through to show you some examples and hopefully give you an understanding of how the tool works. So let's first of all build a network. I'm going to click on an example set of proteins. These are standard names in here. You don't want to put in more than about 10 because of the amount of data that the tool has to pull together. You won't be able to sort of visualize it very well. But here we've got an example of 10, and what we can do is then choose a tissue of interest that we want to query that data set in. So if I pop up this box, we've got options in here from the Illumina body map data and from the GTX data sets. These are all gene expression data sets. So we can select one, or we can use this little search box up here. Let's look for kidney data. You'll see it dynamically changes. What we'll do in this example is we'll choose kidney from Illumina body map and submit that to the tool. So it's retrieving expression level data from that kidney data set, and then those genes which are expressed, the proteins are being explored for protein-protein interaction data and presenting you with this graphical network of all the interactions of your original search terms, which are highlighted in green, the green nodes, and shown up here along the top in green. And the blue nodes that they are interacting with, which are also expressed in the tissue of your interest. The nodes are scaled based on expression values within the tissue that you selected, so some are large, some are small. You can quite easily see the expression level scaling. If we hover over a node, as you just saw, we get more information about that node. So this is the network pane on the left. On the right is what's called the data pane. It defaults to showing a help file that gives you information about what's going on your screen. But if we want to explore one of these nodes in more detail, let's pick this central node and click on it. We can see it highlights all the connections. The node changes color, but it also appears in this box in the top right hand corner of your screen. If you click the info button, we load annotation data about that node from various sources into the data pane. We've got names, we've got sequence, we've got functional data based on reactome pathways. These all click and link out to reactome. We've got its gene ontology terms to give you a bit more of a functional view. We've got structural data if it's available, so protein three-dimensional structures. If we click on those, we get a larger view, and we can jump out to a three-dimensional viewer at PDBE if you want to look at that in more detail. So we got quite a lot of information in one view. I'll just mention the edges as well. If we look at the edges on this graph, they represent protein-protein interactions from intact data between the two nodes. Where we see an edge is thicker, like this one here, if we hover over it, we can see that there are reactome pathways shared between the two nodes that the edge connects. So we're getting a visual view of where there may be a relationship based on functional interaction as well as just PPI data from intact. Okay, so before I show you the two boxes at the bottom, we're going to start again and do a small network. This time I'm just going to enter a single search term, and again we will select a tissue of interest. We're going to go with kidney again and load that network, and this time you'll see it's a much smaller network, just two nodes, SPP1 as an interaction partner. We can, as usual, click on that node, load its data into the data pane, see information about it. But what we can do is start to expand this network around this new node. So now it's highlighted. If we click the expand around the selected node button in the bottom left, we've now searched for both CD44 and SPP1 as our search terms. They're highlighted up here in green. And we're getting interaction partners for SPP1. So this is in kidney from Illumina still, Illumina body map. If we want to explore this in a different tissue, we can pop up the box. Let's choose kidney from GTX. So we can click change tissue and it will rebuild the network for the new tissue, which has changed up here to cortex of kidney. We've still got the same nodes as we had before. If we look in a different tissue, let's say lung, and rebuild it, here we can see we've actually got an extra node appear. This is another interaction partner of CD44. The relative expression levels of the nodes have changed, indicating the expression levels are different in this particular tissue. If we change again and select skeletal muscle, we'll see that actually our other nodes now disappear. Only CD44 is expressed in this skeletal muscle network. So let's go back to kidney where we started, and I'll show you again. We can continue to build this network if we want to expand around another node. We can continue to do so. We can carry on building our network as much as we want to and changing the tissues of interest to whatever we want to explore, and always being able to highlight a node and load more information about it. So hopefully that's a quick introduction to help you get started with using the tool. If you find it useful, we'd really like to know. If you've got any suggestions for information that you'd like to see or changes to the user interface, we'd quite like to know that too. We've only just released it, but it's fully functional, and hopefully will give you a view of your data that you'll find useful and informative.